zoonotic disease and novel approach in wound control. I am extremely happy and thankful to our chief guest of the webinar, Professor J.P. Sharma sir, Worthy Vice Chancellor, Sri Kashmir University of Agricultural Science and Technology of Jammu, for his permission and valuable advice to conduct the webinar. Due to some urgent engagement, sir will not be join us now and probably he is join later. Hearty welcome to Professor Rajesh Katos, Dean Student Welfare, Head of the Department, Veterinary Parasitology, Dean M.S. Barwal. I cordially welcome to eminent speaker, Dr. Chandrawathani, ex-Director of Research and Innovation and Chief Editor of Tropical Biomedicine, Malaysia, and Dr. Reuben, Professor Veterinary Laboratory Diagnostics, University Putra, Malaysia, and all the participants. Welcome once again. Now, may I request Professor Katos to inaugurate the webinar with your wisdom of word. Over to Dr. Katos.
Good morning to all of you. I am audible to everybody?
Yes, sir. Yes, sir.
Okay, so today's topic, zoonotic diseases and novel approaches in worm control. Both are important, as Jammu Kashmir is concerned. Slowly in India, we are heading towards the nuclear families. People are keeping pets, but the disposal of feces, no proper methodology is existing in our state. So upcoming diseases will be from dog and cat borne, because when we have examined dogs and cats in this region, large number of parasites have been observed which have a zoonotic potential. So in that way, one of our team under Dr. Anish worked on zoonotic parasite. He worked on Cryptosporidium, and we could find both the lineages from animal to human and human to human lineages were available at this place. And then presently he is working on the fly diagnostic test, which is causing immense loss to goats in Jammu and Kashmir, because Jammu and Kashmir have roughly 45 million small ruminants, and these animals suffer with wide array of diseases. So I am working on the epidemiology of the gastrointestinal helminths. He is working on the diagnostic feature for the warble fly, which is working, and probably they will develop a diagnostic kit for it. Dr. Shanku, she is working on maggot deregiment therapy. Her team is working on that. We have already taken 2-3 flies where we have worked upon, and we have got very good results in diabetic and healthy individuals. So Dr. Godara, the third teacher from this department, he is working on acaricidal resistance. His team is working in all the districts of Jammu and Kashmir. We have taken all the chemicals which are acaricides which are being used, and we have documented the level of resistance against different acaricides and susceptible chemicals available in those districts, and it has been given to the line departments. I want to say 2 things only, because zoonotic diseases, one of our experts will be delivering zoonotic diseases, the other madam will be delivering on novel approaches in worm control. Just I want to say 2 words only. First, it is my heartiest welcome to both the orators and all to the listeners. The important thing I want to say, because we have seen that inflammatory disorders are very less in the developing nations, because there the parasitic infections exist in low-income countries, and high level of parasitic infection is there. This is an established fact further that there is a reciprocal link between the endemic helminthic infection and reduced prevalence of allergic reactivity and autoimmune antibodies. Hygiene theory, everybody knows about it. But as we give deworms, the same individual will react to the same antigen earlier, which was not reacting to because of the parasite, because the helminths, they immunomodulate the response. The down modulation of these immune dysfunctions depends upon the parasitic species and intensity. In COVID, what we are seeing, in COVID, the response is, inflammatory response is much stronger in the developed nations, whereas in developing nations, the inflammatory response is low. So we can exploit these factors in helminths or their byproducts can be tried in therapies. But here in India, we are giving ivermectin to the patient, and it is giving good results. Uh, then I went into it. I went into the literature. Ivermectin stops the replication of the virus. But if it removes the parasite, then the inflammation will be more stronger. So two contradictions are going together. So I want to say we must see that parasites have co-evolved, and we must see into it how it works and how it modulates the response or inflammatory response, which is reduced in the individuals who are suffering with the parasitic infection. With these words, I can say I welcome from core of my heart both the speakers and all the listeners. I pass on the mic to Dr. Shankar again. Thank you.
Thank you for your word of wisdom. Now, dear friends and participants, we have with us our leading scientist from India. He will give a remarkable lecture on status of parasitic diseases in India. He is nothing but our Professor J. Rao sir. He was a former head of the department from Indian Veterinary Research Institute. Let's listen to him. Aman? Hello, Aman. Aman? Amandeep? Actually, sir was sending a video recording. So just wait.
He is making
Maybe
it to run.
maybe technical problem. Yes,
Yeah,
it
yeah.
is present.
Hello, everyone. Thank you
It's
so
not
much
visible.
for having me for today's web event. I'm indeed delighted to be here and sharing my views through this international webinar hosted by the Faculty of Veterinary Sciences and Animal Husbandry of the Sher-e-Kashmir University of Agricultural Sciences and Technology, Jammu. Designed as a virtual conference featuring two diverse areas of topical interest, one on zoonotic diseases and the second on novel approaches to worm control. I'm sure the two foreign speakers will unravel the science and the knocking trends for useful deliberations on the subjects. I'm also certain that the learned university faculty led by Professor Katoch will also be on hand to take the story forward and provide inputs. As you're all well aware, the former state of Jammu and Kashmir and presently the Union Territory has been regarded as a heaven on earth and is also called the biomass state of India. Dr. M.S. Swaminathan, the visionary former Director General ICAR, once famously said, and I quote, "If agriculture goes wrong, nothing else goes right," unquote. With a rich biological diversity and then people that are traditionally dependent on biomass-based resources. Perhaps this premise may not be very apt to this region, if I may say so. I have in fact no qualms in stating this. At the same time, it is gratifying to say that the subsistence requirements of the people for food grains, firewood and fodder, timber for housing, food, milk and medicines are all derived from the plant and animal resources available in the state. The state is a home to about 75 species of mammals, besides several subspecies belonging to 54 genera, 21 families, and 8 orders from a biological systematics point of view. Carnivores represent 32% of the total mammalian fauna in the state, while the nuclear families in the state have a passion for companion animals. Among the livestock, large ruminants and the multifunctional small ruminants reared under intensive system contribute significantly to the food basket, being the dependable bank on hooves in times of need. Whereas in mountainous areas, large numbers are kept under nomadic regime and animals are well adapted to a variety of climatic, parasitic and husbandry conditions. It then goes without saying that the parasite being ubiquitous are a major factor limiting the animal production and their welfare. In addition to the impact on animal health and production, control measures are costly and often time consuming. A major concern is the development of resistance by worms to many of the common chemical agents used to control them. Given the background, one of the ways in which the livestock production can be increased is by reduction of losses due to diseases and particularly the morbidity due to the parasitic diseases. Then the next question that arises is, are we prepared for this persistent problem of worm control, both in parasitic
I think it's not audible. Is it audible, sir?
At present, it is not audible. Earlier, it was slightly audible, but the sound was not up to the mark. Whether the presentation has been finished?
No, sir. It's going on.
But it is not audible.
Yes, sir.
Should I convey to him?
possible today
Yes,
by
sir.
Now it has come.
preemptive treatments,
Yes.
parasite monitoring
Actually,
schedules,
everybody has to
and
mute.
non-chemical strategies such as nutrition, genetics, pasture management, and so on and so forth. The control strategy of mass drug administration is often suboptimal. This comes largely from the many observational studies undertaken over the years. Hence, there is a need for a workable vaccine against important gastrointestinal worms. Then, coming to the other topic of our discussion about zoonotic diseases or illnesses associated with animal contact, it must be said at the outset that nearly 8 months into the unprecedented global COVID 19 pandemic, we are yet to come to grips with the public health emergency created by this novel coronavirus. The current outbreak has been given a fresh impetus to the global efforts to raise awareness on the zoonosis, and the present webinar is a small step in this direction, I consider. As you know, there are about 150 zoonotic diseases which are known to exist. According to United Nations Environment Program, about 75% of all emerging infectious diseases are zoonotic in humans. Further, it is also well documented that 90% of the recorded human diseases in the last 40 years or so have had their origin in animals. You all know that SARS-CoV-2 virus has a putative animal origin. As vets, we can all appreciate better that pet ownership can have health, emotional, and social benefits. At the same time, these pets can serve as a potential source of zoonotic pathogens. According to WHO, there is a coexistence of humans in a complex, interdependent relationship with the companion, production, and wild animals. We depend on these animals for our food, livelihoods, and well-being, as well as with the environment we live in. A close look at the Indian scenario on the parasitic zoonosis suggests that prevalence of zoonotic parasite is an understatement of the problem. This is largely due to the lack of proper surveillance and the shortage of information about the existence of asymptomatic animal carriers. No doubt, they are prevalent throughout the country at varying rates. However, emergence of diseases such as human echinococcosis, hydatidosis, neurocysticercosis, cryptosporidiosis, and toxoplasmosis in those with acquired immunodeficiency syndrome, along with the re-emergence of diseases like the cutaneous leishmaniasis, Japanese encephalitis, leptospirosis, etc., poses a serious threat today. The prevention and control of these neglected parasitic zoonoses, understandably, is a great challenge. It calls for effective periodic surveillance, forecasting of zoonotic diseases, and capacity building of the competent workforce. In today's time, the main reason for the spread of this impending new zoonotic pathogens is through direct contact with the live poultry, the rodents, the reptiles, the insects, and wild animals. Generally speaking, transmission of zoonotic diseases from animals is primarily by direct contact or ingestion or indirect contact with insect vectors and contaminated inanimate objects, and also by inhalation of aerosolized materials. Importantly, we must realize that with the expansion of agriculture and livestock farming, this is in fact disturbing the natural ecosystem of wild animals, thereby increasing the burden of zoonotic diseases in the population. The current COVID pandemic, in fact, mirrors the broken relationship between the humans and the Mother Nature. According to new study estimates, after the current pandemic, there may be spike of malaria, tuberculosis, and HIV, and many more. This is yet another challenge to all concerned in the public health system. We also cannot overlook the occupational risks due to zoonosis to various categories of people, including vets during their veterinary treatment and those engaged in animal-based research. The other major risk points are among the personnel working in slaughterhouses, tanneries, or wool factories, and those dealing with import or export of live animals, etc. Therefore, a key component to successful disease prevention program is ensuring that vulnerable people are made aware of their risks and receive accurate and timely advice on risk mitigation strategies. I like to wrap up the discussion with a remark voiced by many experts that COVID-19 must act as a lesson to stop further such outbreaks in the world. And it can only be achieved by protecting the animals and their natural habitats and ensuring a healthy ecosystem. Finally, I'd like to thank the organizers and wish the participants new insights into the focal theme of this particular webinar. Thank you.
Thank you, sir, for your valuable discussion. Now, may I request our distinguished speaker, Dr. Chandrawathani, to deliver her lecture on the topic Novel Approaches to Worm Control in Small Ruminant. Before that, I am going to read her biodata to introduce her. And those participants wanted to ask, they may write a question in the chat box. Dr. Chandrawathani obtained her DVM from University Partinian, Malaysia in 1985 and subsequently completed her master's specializing in veterinary helminthology in 1988. She started her career in Veterinary Research Institute as veterinary officer in parasitology department and subsequently headed the department in 1994. She further went to obtain her PhD in applied helminthology from Swedish University of Agricultural Sciences in 2004. Returning back, she became the Deputy Director of R&D in Veterinary Research Institute from 2007 to 2015. She had a short stint as Head of the Veterinary Services in National Wildlife Department, followed by appointment as the Director of Research and Innovation in DVS in 2018, spearheading research for the Department of Veterinary Services in various areas of animal health and production, with special interest in parasitology, anthelmintic resistance, and control of helminths in ruminants. Her effort in research and teaching has earned her several national and international awards, namely the World Association for the Advancement of Veterinary Parasitology Honorary Membership Award as the first female and second Asian to receive it in 2019. She has retired from government service and is currently the chief editor of Tropical Biomedicine Journal. Over to Dr. Chandra.
Thank you very much, Dr. Sanku. And ladies and gentlemen, I'm just going to present now. I'm going to entire screen. Okay. So I need your permission to share the screen.
Pardon, ma'am?
I need your permission to share screen. So
No, you just share the screen.
okay, share. Hang on a minute. Okay. My screen hasn't come up yet. Just hang on a minute.
Ma'am, bottom right,
Yes.
you will get dots here.
Yes. 3 dots.
Present now.
Yes. Presenting to everyone, but my screen is not coming up.
Wait.
Maybe takes a bit of time.
You have shared the screen.
Yes, I've shared the screen, but okay. Have you got that?
Just wait. No.
Okay, can you see?
Yes.
Now we can see.
Okay. Can you see? Can you hear me?
You can carry on, ma'am.
Yes, we can hear you, Dr. Chandra.
Okay, thank you. Okay, before I start, I'd just like to say a big thank you to the Honorable Vice Chancellor of the University, Professor J.P. Sharma, and also Dr. J.R. Rao, who gave a very nice talk just now. And a special thanks to the organizing committee with Dr. Sanku Bodkataki, as well as Dr. Rajesh Katoch. These are all my old friends from Jammu Kashmir when I visited some time ago. So I'd like to record my gratitude to all of you for organizing this webinar. In spite of the COVID situation in both our countries and the whole world, we are able to see each other and share our information. So, ladies and gentlemen, it gives me great pleasure to share some of the information that I have collected over the past 30 over years on novel approaches to worm control in small ruminants, especially for the tropics. And I'm sure in many parts of India, you still have a tropical climate and you can utilize this information. And the good thing about research all over the world is we can share with each other and copy what is good for that works. So, ladies and gentlemen, it gives me great pleasure now to share this information. You can see background of this slide.
Sorry,
It's,
ma'am. Sorry, ma'am. I'm interrupting you. Your slide
yes.
is not visible again.
Okay.
Your slide is not visible, ma'am.
Okay, what shall I do?
You do once again.
I can see your slide. I can see my slide. Shall I? And okay, can you see?
Can you repeat the process again now?
Can I repeat the whole process? Sure, I can. Okay, I'll just go here and I will just repeat the whole process.
Okay.
I'm going to present now, entire screen. Okay. Share. Share screen. Okay. And I'm going to my slides. Can you see?
I can see your slides, Dr. Chandra.
Okay, good. Okay, I'm going to go to screen. Okay. Okay, can you see?
I can see you.
Can. Okay, right. So I'm just going on to the next slide. Just let me know if I need to stop at any point. Okay. My sincere thanks to all of you for sharing Kashmir, University of Agricultural Science and Technology of Jammu. And today, what I'm going to share with you is the green solutions for worm control. And what I will share is a basket of options for farmers. And this information is quite useful because we have used it in our country for a lot of the smallholder farmers. And our main concern here is the worms, gastrointestinal worms of Haemonchus, Tricho, Esophagus and Cupiria, which are the strongyles. So I just like to share this slide with you because we are from Malaysia and we are happy that we get to have this connection with your university and colleagues and parasitologists in India. And I would like to always welcome you to Malaysia anytime that you are here, or you can come after the COVID situation. We'd love to have you and have more connection with you. So a lot of my work was done in the Veterinary Research Institute, IPO. In IPO, this is a picture of the institute, and you can Google and see a lot of the research that we have done in there. And over the past 30 over years, the information that I'm going to share with you is from BRI IPO. And I know that India has got a very famous Indian Veterinary Research Institute too. So first of all, I'd like to share this slide. In Malaysia, we have many acres of oil palm plantations. And under the oil palm plantations, you can see it's a beautiful grass here. And this is a perfect environment for keeping sheep or goats or even cattle. And we found that for these gastrointestinal helminths of sheep and goats, we find that the larvae survive for around 4 to 6 weeks under tropical conditions such as this. So, of course, in many of the other tropical countries like in Indonesia, and Thailand, and Cambodia, and even India, the time frame for larval survival may differ a little bit. And over here in Malaysia, our relative humidity is 80 to 100%, which makes it a perfect for the larva. And we have temperatures of about 28 to 32 degrees, and we have rainfall almost every week and every day. So in 1 year, we can get almost 2,000 over mm of rainfall. So with this kind of conditions, I would say it's a perfect environment for larval survival and for gastrointestinal helminths to promote its life cycle. So in Malaysia, over the years of 1989 to 2015, we did several studies on the epidemiology of helminth infections under rubber and oil palm. These two crops are the cash crops in our country, and we have several thousand acres of rubber and oil palm. And this is the kind of environment that you will see under these trees, and you will have goats and sheep grazing under them. And as a result of this, this is a perfect way of setting up a rotational grazing system for controlling Haemonchus contortus. And we take into consideration the microclimate in these plantations. So in Malaysia, we have integrated farming. We have goats and sheep that we can keep over sheds like this. And the pond is for fish, inland fish farming. And we also have lots of tropical fruit trees growing. So this is the kind of a typical farm in Malaysia. And we have also a lot of commercial farms where we keep doppers and other many species of goats and sheep that we bring into the country. And also we have many smallholder backyard farms, which means that the farmer probably has another job. He has some other farming activities as well as a first job, and farming is just a part-time job. And as a result of that, you can see the sheds will be very simple, and they may be keeping about 20 animals. Like in this shed, he will probably keep 10 or 15 animals. And these are called smallholder backyard farms. And to facilitate these smallholder backyard farms and convert them to commercial enterprises, one of the biggest challenges we have is to control parasitic diseases. So we also encourage farmers to give supplementary sources of feed, like from here you can see some branches of plants that they are hanging here for the goats, and they also give a pellet feed. And they keep the salt in this bamboo hanging here for the goats to lick on the salt. So this is a typical Malaysian scenario here. And you can see that the feces that is collected under the sheds is all piled up around the farm. So you can imagine if this feces was having a lot of eggs, gastrointestinal helminth eggs, they would hatch and they would contaminate all the pasture around here and all the grasses around here. So this constitutes an important infective area for the goats and sheep. So hemoncosis as a result of this is an important challenge to small ruminant farming. And here you can see the hemoncus, which is a typical barber pole worm. You can see the red and white stripes. So I would say that the laboratory and post-mortem diagnosis is very important, very crucial when we want to control helminthiasis in sheep and goats. And another thing is, after the diagnosis, we need the drug therapy. So this is traditionally, this has been the method of controlling the worms. We do a lab analysis of master egg count with the feces of flotation. And then if the animal is dead, we do a post-mortem. Once we find it is hemoncus, we go straight to the anthelmintic therapy. This is what has been practiced over the past 40 or 50 years. But as time goes on, we find that the worms are becoming resistant to the drugs that we use. Okay, so my talk today is about how do we control it if we cannot use drugs. So I just give you a little bit of history about our anthelmintic resistance status in Malaysia. The first case was reported in 1993, and over the 10 years, we succumbed to very severe anthelmintic resistance in many of the farms, meaning the farms could not use anthelmintics as freely as they used many years ago. Okay, so the anthelmintics were not working or not effective for the goats and sheep. So what are farmers going to do? So that is the question that we had to answer. So a little bit of background about the worms that we are working with. The question is, should we continue to use drugs when they are not working? Okay, so what is anthelmintic resistance? And we had anthelmintic resistance to macrocyclic lactones, which are ivermectins, benzimidazoles, levamisols, AADs, and the Quantels. And these drugs are available all over the world, and I'm sure in India too, and many parts of the world, these drugs cannot be used. One of the oldest drugs is benzimidazole. So what happens is when the sheep lay eggs and they hatch into larval stages and reinfect other sheep. And when you give the treatment, it's not working. Okay, so before we try to control the worm, we have to know the life cycle, and then we can expect to control the worm because we cut the life cycle in certain places. So in the Malaysian, one of the typical scenario in Malaysian farms, for example, is like this. We have oxfendazole, levamisole, moxidectin, and clozental, and we do this fecal egg count reduction test, and we found resistance to these drugs. And in some farms, you can see only one drug can be used. So this is the typical scenario in more than 60% of the farms over the past few years that we surveyed. And the worm population in the farm is basically Haemonchus. So Haemonchus contortus, as you know, is a bloodsucker, causes severe anemia, and also it gives rise to, allows other infections to come in. So as a result of the poor immunity, the animal will be susceptible to pasteurellosis and other bacterial or viral diseases. So we have a small percentage of the animals also have Copiria and Trichostrongylus. So if you remember our drugs that we can use for Haemonchus, Clozental is very good because it works on bloodsuckers. So the other important thing that we'd like to share here is that most of the worm larvae on the grass is found about 5 cm off the ground. Okay, so they move up and down the grass. You will not see it on the higher up here, on the high 15 centimeters or 10 centimeters, very little. Most of them will be around here. So if you remember the photo that I showed just now under the oil palm and rubber trees, the grasses are very short. So it's perfect for the larva to go up and down here. Okay, this is where most of the larva will stay. So once we know the character of the larva, we can control them. So the grass under the oil palm trees are short, the environment is damp, and is perfect for larval survival. And helminthiasis is very common in a lot of our sheep and goats. Now, what are the risk factors for anthelmintic resistance? Basically, the main factors would be high treatment frequency. That means the farm has been using a lot of drugs more than 6 times a year, and they are probably underdosing. How are they underdosing? Because they do not weigh the animal. They are giving a visual weight appraisal. That means by looking at the animal, the farmer is probably guessing the weight. So most of the time they underdose. And also another factor is the farmer is trying to save cost, and so he may underdose. The use of same anthelmintic class over and over several years, that will make cause anthelmintic resistance. And of course, poor pasture management, which means we allow our sheep and goats to graze on pastures, permanent pastures. So the estimation of anthelmintic resistance is using the fecal egg count reduction test, which takes 2 weeks. This is a long time for a test. And you can also use larval development assay or egg hatch assay, and also molecular tools nowadays. So this is important for us to develop these tests so that the farmer can get the information as soon as possible. So the worm control should be a combination of techniques such as body condition scoring, fecal egg count detection, larval detection, and a pharmachar, the use of pharmachar. I'm sure all of you know about pharmachar. It is a card like this in the background. You can see it's a card, and you can assess the anemia, level of anemia of the animal, and you can treat individually. You do not have to give a blanket treatment to all the animals. So for farmers who have poor resources, what are the alternatives that they can use? I would say that these are tools that we can use to help farmer identify worm infections and then give the medication or take some steps to reduce the worm burdens. So I would say these techniques are underutilized, and I would say that it's important to share this information. What is pharmachar? What is fecal egg count detection? And what is body condition scoring? It's very important to share this with farmers so that they can use this as a tool. And farmers need a constant awareness on proper use of anthelmintics. If you, in some countries, I would say farmer is reluctant to use anthelmintics because it's expensive. But in some countries, anthelmintics are given free of charge. So this will accelerate anthelmintic resistance. Pasture management is another tool that we can share with farmers. And also when they purchase and transport livestock from one area to another, to be aware of the worm conditions. So now coming to the alternative worm control methods. If a farmer does not want to use drugs, does not want to use anthelmintics, what can he use? He can use rotational grazing. Okay, now you see in this picture, there is a fencing here and all the sheep are on one side. So after they finish the grass here, they are moved to the next paddock. So the quality and quantity of the forage is very important here. If you put them on a pasture that does not have any grass, then you're going to affect the production of the productivity of the animals. So here's another picture where you can consider using high sheep density for a short period of time as part of tactical grazing approach, meaning you put them on one paddock, after the grass is finished, you move them to the next paddock. And so because of this, you need many paddocks and a large area. To save cost, you can probably use temporary fencing to save cost. So this fencing can be moved when the animals want to be moved. So in Malaysia, we did a big project with Ian Barger and team under the ASEA program. And in this program, we had several countries, Indonesia, Thailand, and India also trying out this rapid rotational grazing system. And we found that you can put animals 3 to 4 days per paddock. Why 3 to 4 days? Because they would have released the feces in this paddock and it takes 3 to 4 days to hatch. So before they can consume the larval stages, they are moved to the next paddock. And they will come back to the same paddock after 30 days when the larval load on this paddock has reduced substantially. So in rapid rotational grazing, you only do 2 initial treatments to clear the animals of any existing parasites. And another trick that we used here is we do a Monday-Thursday program where we move animals every Monday and Thursday. So it's easy for the farmer to remember. It's Monday today, we have to move paddocks. So the only drawback for this system is you have to have several paddocks, and you must have the area, the land area, and the good quality grass. So here we have rotational grazing, and you see here each paddock is about an acre, and you can calculate according to the amount of grass you have, how many animals you want to put in here, and then you move them every Monday, and on Thursday you move again, and Monday again. So they come back to the original paddock after about 30 days. But here it's 10 days per paddock, which also comes to 30 days. And we can somewhat help to kill off some of the parasites before the goats return here. Okay. Another method that can reduce worm burdens in sheep and goats. In the Pacific islands such as Fiji and Samoa, the farmers are smallholders and they use tethering. Tethering is a good way of controlling worm burdens also, because you can tether today at this tree and tomorrow in another tree. So there is some kind of a pasture rotation. The only problem is you must have enough foraging area, and it should be protected from adverse weather and predators. Because it's tied to the tree, the goat cannot run away if there's a predator, and it is isolated from companions. So this is another thing that we are concerned about nowadays, about animal welfare rules should pertain. So here again, you have tethering in some of the Pacific islands. This is a common way of farming goats and sheep. So for cut and carry, the other method of reducing worm burdens is zero grazing. As you know, the larva is always on the grass. So zero grazing means you are collecting the grass, cut the grass from areas which is not grazed by animals. So then the grass is clean, free from larva, and you can cut the grass and feed the animals. We in Malaysia we have oil palm fronds. That is the leaves, and this is the way we cut the leaves, and it's chopped up and we give it to the animals. So this cuts the life cycle of helminths. That's one of the ways. So oil palm fronds are a byproduct in Malaysia and probably in some of the Southeast Asian countries where palm oil farms are grown on large acreages. You will find that oil palm fronds is about 5,500 kilograms per hectare per year. So this is a good byproduct. It has a fairly good crude protein, and also it is a low cost byproduct. So in Malaysia, we encourage farmers who have oil palm, acres of oil palm plantations to utilize this. And here you can see how they chop the oil palm fronds, and this is how it looks like, and it's fed to the animals. So other local byproducts that we give can be some of the legume leaves, etc., mulberry leaves, and all that. So here's another agricultural byproduct in Malaysia. In some parts, we have large acreages of tapioca. Tapioca is used for industrial starch process production. So when they harvest the tapioca tubers, you get a lot of leaves being wasted, and this is another byproduct that we can give the sheep and goats. But we have one drawback here is that it has a cyanogenic compounds in leaves. So we have to chop the leaves, or it should be wilted and chopped. Then the cyanogenic compound is evaporated and it's safe for the animals to eat. But these are just additional feeds. So we also found that feeding cassava leaves has got some effect on the total number of worms in the gut, gastrointestinal tract. So there was a significant drop in the abomasal worms, even in the small intestine, and large intestine it didn't affect very much. So this is another positive effect that we saw with the cassava leaves. Now, coming to herbal remedies, I think India has no shortage of herbal remedies, and in Malaysia, we were looking at the products or the plants that we have in Malaysia and looking at the anecdotal evidences that we have and decided to try them out. So neem is one of the important plants. It's the same neem probably that you have in India, and the parasites was significantly higher in control group, meaning that when you do not give the leaves, then there was more parasites. And they had an effect on worm numbers in sheep, but not on the egg counts. So this is something that we need to investigate further. So this is one of the papers that was written by my group. In 2013, we did some studies on neem decoction and neem water extract studies. How do we test for plants with anthelmintic activity? If you suspect a plant with some anthelmintic activity, how do you do it? It's an in vitro screening of herbal plants. And basically, it is measuring the inhibiting activity of plants on the infective stage, which is the L3. And in vitro screening is a cheaper method rather than you are using animal models. And the plants are chosen based on literature review and anecdotal surveys. So this is a part of the work that we did in plant extract preparation. We sometimes have to crush them, grind them, and then we sieve them, and then we have to freeze dry, and then we try it on the larval stages. So we also tried this larval migration inhibition assay on papaya. I'm sure you have it in India too. Papaya is, we also can eat this fruit, and betel leaf and neem leaves. So the LMI showed that neem leaves and betel leaves has some promise of knocking out the larval stages. We also tried with bitter gourd and a noni leaf. So this is a noni fruit, and the leaf is the one that we tried with, and also had some promising effects, but we need to do further studies. As you know, all the studies on herbal candidates tested by LMI, and further needs to be tested on goats, on live goats. And we did a lot of these studies in a government farm that was very near to the Veterinary Research Institute. And this can be quite an expensive affair because we need to use live goats. We need to probably post-mortem them. So many of the studies is ongoing. Another way of controlling worms is the effective microbes. I'm sure you know about effective microbes, which is basically a lactobacillus soup with molasses and a fruit base. And what we did is, we being in the tropical country, we have lots of fruits that are going to rot and waste, and so we can use this to produce effective microbe solution. This is a fluid and a feed additive. So this is how it looks like. It's like a juice, and basically using anaerobic microorganisms in a carbohydrate-rich liquid carrier with a molasses. So we found that using this on a daily basis even can reduce egg counts by 80%. So this is good. This is not dangerous. It is not a drug. It is easily available and can be cheaply produced. And this is how we mix it in water, drinking water, 1 to 2,500, and body spray for ticks, it can be used, and also for composting, for fertilizer, composting the feces. So this is, has some promise. So one more that we like to share here is the feeding medicated urea molasses block. And these studies were done also with the Australian Agriculture Research Grant. And what we did is, all the sheep and goats, we have to give them a mineral block. And we incorporated the medication into the mineral block. So when I say medication, it is probably benzimidazole powder, and the amount is very small. So even if that farm was experiencing anthelmintic resistance, we can use this. Basically, it knocks out the larval stages that get into the animal. So it also has multi-nutrient to help enhance the nutrition of the animal. So urea molasses blocks are used in several countries. So it's very easy to have incorporate a small amount of drugs inside. This was quite highly effective in reducing fecal egg counts in 90%, more than 90%. So the cost-benefit ratios for using medicated blocks is 1:1.3 and 1:7 across several countries. So this study was done in several countries, and the income increased by 33% to more than 400%. So it was done in several countries here, as you can see. So Malaysia is one of the countries that participated in this trial. And here I'd like to share again about Pharmacha. And Pharmacha, this was first introduced by South African scientists and parasitologists. And I think this is very good for smallholder farmers who are having a small number of animals, because they are able to check the mucous membranes of each animal, and they compare the color chart, and they can treat animals individually. So if you really cannot do rotational grazing or any of the other control options and have to use a drug, then this would be a good option to try. And for biological control, I would like to add here that in the early 2000s, I worked on this biological control using Duddingtonia spores. So this technology is introduced by the Danish parasitologists, and at that time, the spores were being produced in Denmark and Sweden and used for worm control in horses as well as small ruminants. But we could not import those spores to Malaysia because it was deemed quite dangerous. So what we did is we decided to look for Duddingtonia spores in Malaysia, and we found Duddingtonia is very commonly available in feces of sheep and goats. It's commonly. So we decided to grow it and to produce our own Duddingtonia spores for biological control. So here I share with you this spore is a Malaysian Duddingtonia. It reduces pasture contamination if fed on a daily basis. So this is where the trick comes. Biological control was the work done over early 2000s. And here you can see us growing and looking for the Duddingtonia. Once we found the Duddingtonia, we can grow a lot of it and get the spores and put it into feed and feed the animals. So Duddingtonia needs to be fed every day, and that's where the challenge comes, because to keep Duddingtonia spores in our hot, wet tropical climate, its spores are constantly growing and sporulating. So we put it into feed blocks, the spores, and the animal, when it goes out grazing, comes back and it will consume the spores. The spores will go into the animal and comes out with the feces. And when the larva hatch, the spores will also grow and trap the larva. So that's how it works. And just want to share with you the precautions that we need to take when we want to, if you definitely need to drench with some drugs. In order to maintain the use of your drugs for a longer period of time, or the effective of your drugs, this is what you need to do. You have to drench properly and correctly. And this is something that we share with farmers. First of all, dose the heaviest sheep or goat in the group. It's very dangerous to do a visual appraisal because sometimes we can underestimate the weight. And always to drench gun should be calibrated so that the correct amount goes in, and put the nozzle over the tongue so that the drug goes into the GI tract properly. Otherwise, it gets lost and it will bypass the rumen. So this is how to drench a goat over the tongue. Okay, so that's an important part of worm control that we have to constantly remind our farmers. Another important points that I'd like to share here is withholding food. So we found that when you withhold food, something like fasting, 12 to 24 hours before treatment, it makes the drug more effective. Okay, and also the storage of the drug should not be in direct sunlight and must be in a cool, dry environment. So an expiry date must be checked all the time. And another thing that farmers always forget is shake the, especially BZ products, Check it before use. Otherwise, you get a sediment at the bottom and the drug is not really the right concentration. Okay, so the limitations that we have, that we face, I'm just sharing some of the experiences that we have here. And there's a need for resistant breeds of sheep and goats to worms. So there are many research groups in parasitology. They are working on resistant breeds together with breeders, and that's very good, I think. And upgrading diagnostic services in Asia. The very simple fecal count, like master fecal count or flotation test, should be widely used because it is very cheap. And awareness of novel approaches to worm control in Southeast Asia can be shared with all our counterparts, especially with farmers. And we need, over time with the climate change, we need epidemiological data, anthelmintic resistance data, and farmer chart. How effective is it? So these are possible areas of research that is directly useful for farmers. And we need research in coming up with test kits. And this slide I have shared with several parasitology groups because I think as parasitologists we should communicate with each other and support each other in coming up with these test kits or information on how parasites affect growth. And the extension info is very important. And also on pathology of helminths in livestock, the unseen effects. And this info should be shared with farmers. And nowadays with the new technology, it's important to have apps that are going to help us. Test methods and biochemical tests that make things testing faster and more efficient. So continuous training and sharing is something that I like to promote because to carry out research is an expensive affair. Once we have the results, it's very important to share with all our colleagues all over the world so that we can extrapolate that information and use it in our part of the world. Okay, I like to share some of the work that we have done with our Cambodian colleagues, and this is how it looks like. Also hot, wet, tropical climate, and this is how the sheds look like. And they have laboratory tests and information on nutrition and breeds that is suitable for their country. And in Thailand also we have raised floor farms like this, and they use a lot of byproducts, agriculture byproducts, something similar to Malaysia, and they have a good export industry. And in Indonesia, similarly, we have goats and sheep kept in sheds, and they have laboratories. These are the laboratory in Medan that we visited. And in Philippines, of course, they are on the way to commercialization. They have big dairy farms, just like in India. I'm sure goats and sheep farming is very well established. And this is in Malaysia, some of the basic laboratory training that we provide for our laboratory staff. This is total worm count that we do, and looking at for worms in the rumen and reticulum and the abomasum. And this is one of our students looking for worms. So worm identification, worm counts, larva IDs, important part of parasitology. And I would like to see this being utilized more because this is cheap and easy to do. So ladies and gentlemen, we come to the end of our presentation. Basically all the work that we do for the parasitology is towards safe food for the public. We need to give our public the quality that they require. And I'd like to thank my Department of Veterinary Services and especially the Veterinary Research Institute for allowing me to conduct all this work that I can share with the rest of the world. So ladies and gentlemen, before I forget, I'd like to record my thanks for VRI Malaysia, DVS Malaysia, the universities that have come up with students and the parasitologists who have contributed Malaysian Society of Parasitology and Tropical Medicine that has encouraged parasitologists in Malaysia, and international counterparts who have given so much of information, help, and funding to us, especially the ILPA program, DVS, ASIA, JICA, and IAEA. Ladies and gentlemen, with this, I end my presentation.
Thank you very much, ma'am, for your valuable speech, and thank you very much also for your sparing time with us. Thank you once again.
Thank you. Thank you so much.
May I request Dr. Anish, if is there any questions in the chat box, please read the questions, one or two.
Yeah, yeah. Dr. Chandra ma'am, sincere thanks for enlightening us about your experiences for sustainable control of GIT nematodiasis in small ruminants. Your approach for using alternative measures for sustainable control of GIT nematodiasis against GIT nematodiasis is really very good, and your suggestions that the local feed and the local resources should be used for controlling these infections. Definitely, this is the area where the new work can be carried out in different parts of the country, depending on our regional bioresources. Ma'am, your talk will help us in guiding and devising the roadmap for control of worms in small ruminants. So we are very thankful to you for presenting so nice talk to this webinar.
Thank you. You're
Any
most
question?
welcome.
It's our pleasure, ma'am. Yeah. Any question?
Is there any resistance report of Dudingtonia? Someone think.
Yeah, for Dudingtonia flagrans, you see, we are using the spores. The spore is a physical trapping of the larva, so you cannot get resistance to that. So actually, that's the alternative for the anthelmintic resistance that the Danish parasitologists came up with. Unfortunately, for our tropical climate, the spores tend to start growing faster than we can use them. So we have to keep them in a refrigerator, and the product that we have to produce takes time and money. And so we are not using biological control, but there are some parts of Scandinavia that use that successfully.
Thank you very much, ma'am.
You're welcome.
Ma'am, regarding urea molasses block,
Yes.
What's your opinion? Is it? I think it's very beneficial because we can use easily. The farmer can use easily this. What's your opinion?
Yes, I think it's very good. In fact, the farmer can make his own urea molasses block if he can buy the ingredients and make them himself. In fact, we have many workshops training farmers to do that, because our grasses tend to be poor quality because of the hot climate here. So the urea molasses blocks actually gives the added nutritional value, and that gives the better productivity, and farmers get better profits from that. And also controls the worm burden a little bit, you know. So, and you add the medication inside that, that's even better. So I think it's easy to do and it's easy to apply. Farmers can always do that.
Thank you very much, ma'am.
Okay, ma'am.
Thank you.
Thank you, ma'am, for gracing the occasion.
Thank you. Most welcome.
Yeah, ma'am.
Namaste.
Over to Sanku, ma'am. Namaskar, ma'am.
Now, may I request our next speaker, Dr. Reuben, to deliver his lecture on the topic Wildlife as a Reservoir of Zoonotic Disease in Asia, with an update of zoonotic primate malaria. Before that, I am reading Reuben sir's biodata for your introduction. Dr. Reuben Sharma graduated with Doctor of Veterinary Medicine degree from University Putra, Malaysia, and subsequently pursued Masters in Veterinary Science in wildlife medicine at the same university. He was later awarded a PhD from the University of Cambridge, UK, focusing on molecular genetics and phylogenomics of hemoparasite. Reuben has been appointed as advisor to the World Health Organization, is director on the executive committee of World Association for the Advancement of Veterinary Parasitology, and an affiliate researcher at the University of Glasgow, UK. He has also acted as consultant expert resource person to various NGO and government organizations, including the Worldwide Fund for Nature, the Department of Wildlife and National Park of Malaysia, Department of Veterinary Science Malaysia, and the Ministry of Health Malaysia. Reuben has served as President of Malaysian Society of Parasitology and Tropical Medicine, is the Vice President of Veterinary Association Malaysia, and a member of National Wildlife Genetic Advisory Board and Country Coordinator for the Sakura Science Program, Japan. May I request Dr. Reuben to present his lecture. Over to Dr. Reuben.
A very good afternoon or morning, everyone. And thank you very much, Professor Sanku, for inviting me to share a little bit on zoonotic infections in Asia. I would like to thank SKUST Jammu for inviting me to present today, including the Honorable Vice Chancellor and the Deans of the faculty. So thank you very much for having me. And I would like to share my screen now for my presentation. Please do let me know if you can see my screen, or if you cannot see my screen, or if you cannot hear me properly. Can everybody hear me well?
No, sir, not coming.
But can you hear me?
Yes, sir. You are
Okay,
audible.
great. Okay. Maybe it might take a little while.
Right.
Maybe it's taking some time to load. Huh?
No problem, sir. You take your time.
Yeah. Can you all see my screen?
Yes, can see very clear.
Okay, great. Thank you very much. So, if it takes a longer time to load or anything like that, please let me know, okay? Or if I'm speaking too fast. So, again, thank you very much. And I would like to share with you a little bit of information on how wild animals can act as reservoirs of zoonotic diseases in Asia. And then for the second part of my talk, I would like to introduce to you about zoonotic primate malaria and give you some latest update on the infections which are currently happening in Southeast Asia. Okay, emerging infectious diseases are very important because we know that about 60% of the human EIDs are zoonosis. And out of these, it is estimated that about 72% are from wildlife. So essentially, when we talk about human infectious zoonosis, we must understand the role that the wild animals will play in the pandemic and in the dynamics of transmission of these infections. And of course, there are many pathogens which are responsible, including the bacteria, the viruses, the parasites, and also the fungus. Now, there are many drivers for zoonotic pandemic infections globally. It's a very complex interrelationship with all these factors, but a few of them stand out as very important drivers. One, of course, is agriculture. A lot of land is being converted into agriculture globally, and it's estimated that about 50% of all the emerging zoonosis is due to agricultural development and intensification throughout the world. I would like to focus today on wildlife, which will include overexploitation of wildlife through unsustainable illegal trade, poaching, hunting, and also the trade in wildlife meat, in wildlife body parts as well. And of course, urbanization and industry, which has actually driven a lot of wild animals out of their natural habitat due to habitat loss, destruction of forests, development of industry and land use change has caused a lot of wild animals to be displaced from their natural habitat in the forest, and they're coming closer into the peri-domestic environment, which is very close to human inhabitation. Now, I need to set the stage first when we talk about wildlife infections and how this plays a big role in human zoonosis. We know that there are many hotspots for terrestrial biodiversity throughout the world. We get them throughout the equatorial belt and also in the Mediterranean area. And Southeast Asia, especially, is a great hotspot for biodiversity because we've got very ancient and very old forests, which supports a lot of species diversity as well as population numbers. But sadly enough, although we have a great biodiversity, we are also a hotspot for threatened mammals. A lot of mammals in Asia are being threatened by various things, illegal hunting, poaching, the meat trade, habitat loss, forest destruction, and things like that, because we are a developing nation, a developing region, and we need to develop our industry, and henceforth a lot of new land is being opened, and when we do that, a lot of animals are being displaced from their natural habitat. Now, this is a list, a simple list of the factors that cause animals to decline or go extinct or be threatened. And as you can see, there's a whole list of habitat loss, overexploitation, invasive species, disturbance, pollution, and so on and so forth. But the biggest driver so far is habitat loss. And habitat loss affects all classes of vertebrate animals, including mammals, amphibians, and birds, fish as well, and also a lot of invertebrates, the insects. So habitat loss is a major global issue when we talk about decline of wildlife numbers or displacement of wildlife from their natural habitat. So the big question is, how does habitat loss actually influence zoonotic disease transmission? We find that when there's a loss of natural habitat, the wild animals are displaced from their natural environment and they are forced to encroach into urban and suburban areas, the peri-domestic environment. So they come into this peri-domestic environment and they start to colonize and reproduce very quickly because there's great availability of food resources and shelter, and a lot of these animals, they can subsist on human food waste. So you can find them a lot around areas where sanitation is poor, where garbage disposal is poor. So a lot of this wildlife actually come into the peri-domestic environment. And because these human food waste have got a lot of energy, they are able to utilize this energy and they then start to reproduce very quickly. Now, the thing is that when they come into the peri-domestic environment, they will tend to bring with them the zoonotic pathogens and also the vectors, which were once mainly confined to the forest habitat. Now, when they bring in these forest pathogens, it actually closes the gap, or it narrows the gap in the human-wildlife interface. And when this gap narrows, there's an increased chance of human exposure to zoonotic pathogens that these wild animals can harbor and they can carry, and also exposure to the vectors that come together with this wildlife, especially the ectoparasites and the arthropods. So this is a very simple diagram illustrating the flow of pathogens in the various spheres of transmission. Yeah? So if you look here on the top right hand corner, you've got wild animals or wildlife in their natural forested habitat, and this is called the salvific cycle. Pathogens which cycle in this environment are known as salvific pathogens in the salvific cycle, and every now and then there is a spillover from these wild animals into the peri-domestic wildlife. The peri-domestic wildlife would be things like rodents, rats, bats, birds, small mammals like civets, wildcats, and things like that. And these animals are actually in close contact with their wild counterparts in the forest. In fact, a lot of them actually transverse between the peri-domestic environment and the wildlife habitat as well. And then, of course, wild animals can also transmit infections directly to domestic animals, especially in areas where there is a close contact or there's a bridge between the wild habitat and the domestic habitat, for example, in farms which are close to forests and things like that. And then, of course, humans can also get directly infected through direct contact or via arthropod vectors with wildlife. Humans which spend a lot of time in the forest or those who stay close to forested areas can also get directly infected, or those who go for hunting, or people who consume bush meat or wild animal meat, they can get directly infected as well. So there is a very dynamic flow of pathogens in this biosphere, and therefore, if you want to understand what's going on in terms of emerging pandemics in humans, we need to understand not only the human environment, but we need to know about the peri-domestic environment, the environment that the domestic animals are in, and also the salvific cycle. This is a diagram showing you generally the model of how zoonotic disease spillover can occur. If you look here at the bottom, we've got the salvific cycle of infections in wild animals. And generally, because wild animals have evolved over a very long period of time with their pathogens, what you would see is a general cyclic pattern of infection. Okay, a very general cyclic pattern of infection where you will get in certain times high infection rates or high prevalence in the wild animals, and eventually they will learn to overcome the infection, and the infection will then tend to disappear or be at a very low rate until it goes up back again. So this is maintained constantly in the salvific cycle. But every now and then there will be a spillover to the domestic environment, and these domestic animals act as very efficient amplification hosts. So because they are naive to a lot of the pathogens and they are not natural hosts for these pathogens, so when they get infected, most of the time it becomes a full-blown infection, and they can actually become amplification hosts and even reservoirs of zoonotic infections from the wild animals. Now these domestic animals, because they are reservoirs, there will be eventually a spillover to the human population. And of course, there can also be a spillover from the salvific cycle as well into the human population. And what we tend to find, the trend of infection in humans, is that it starts off very mildly first, but soon as a number of individuals in the population get infected, then it tends to spike up very drastically because we are naive hosts. And eventually, over a period of time, most infections will tend to resolve in the general human population. And this is seen now with our COVID-19 infection, where we get a very high spike in infection rates. And eventually, we would hope to see a decline in infections. And that can be either because there's a vaccine, humans may have some acquired immunity to the pathogen, or the human race is totally wiped out and then there's no more host, and therefore the pathogen dies off as well. So there are many routes of zoonotic infections to humans, either from domestic animals or from wild animals. There are many vectors which will transmit a lot of pathogens, viruses, bacteria, and also a lot of protozoa, especially blood protozoa. Then, of course, you've got aerosols, where we can get infected by droplets, airborne droplets. Oral contamination is very common through the fecal-oral route. Direct contact is also very possible, especially those who are in close contact with animals or animal parts, especially in the wildlife meat trade. You get a lot of animal handlers or wildlife meat butchers getting infected with zoonotic infections. And then, of course, there's formites. But by far, vectors play a very important role in transmission of zoonotic infections to humans. And when we talk about zoonotic infections, it is always very important for us to understand the epidemiology and the drivers in that epidemiological arena. We are very familiar with the epidemiological triad, which is the agent, the host, and environment. But I've included another component, which is the vector, and I like to call it the quartet, because I do feel that agent, host, environment, and vector factors play very important roles in the outcome of infection and also the transmission dynamics of infection. And later on, when I talk about zoonotic malaria, I will be I'll try to address these four factors as well. So as you can see, in the past and even up to now, there are many hotspots, global hotspots of emerging zoonotic diseases in the world. And as you can see, Asia is definitely a very big hotspot there. We've got parts of Europe as well as Equatorial Africa and South America as well. And there are many infections in Asia which we need to be concerned about. SARS was something in the past, but now we've got our other coronavirus, which is the COVID-19 to worry about. Of course, bird flu is ever present. Nipah virus was something very big in Malaysia, and Hendra virus as well in parts of Australia and also in Southeast Asia. So as you can see, there's a very wide distribution of zoonotic infections, and there are actually many zoonotic diseases from wild animals which we need to be concerned about. And as I mentioned earlier, over 70% of the human emerging infections are from wildlife. And honestly, if we ask ourselves today about any emerging human pandemic, the pathogen can almost always be traced to a wild reservoir host or a wild vector. Okay, we talk about West Nile virus, SARS, bird flu, H1N1, Ebola, Nipah virus, Hendra, dengue, rabies, chikungunya, you name it. They always have a wildlife reservoir or a wildlife vector. And of course, now we are talking about our COVID-19 coronavirus with over 31 million cases now, close to 1 million deaths globally. And in the past 14 days, we are seeing emerging foci of infections in South America, in Asia, Central Asia, and also in America. So this is a very big issue now, as we know. There's a lot of speculation that this virus originated from wildlife, not been proven conclusively yet, but there's a lot of evidence pointing towards it. And this infection is here to stay for quite some time. I'm not going to talk today about the COVID-19 disease, because I'm sure that a lot of us have been bombarded with this quite a bit over the past few months, and I'm not going to elaborate a lot on this today. But what I would like to share with you is the importance of the various taxa of wild animals as potential reservoirs and vectors of zoonotic pathogens in Asia. So high on the list are the rodents, the primates, and the bats. You can see that they are potential reservoirs for a lot of helminths, protozoa, rickettsia, bacteria, and of course viruses. So these animals are very high on the list for zoonotic transmission potential. Then of course the reptiles, turtles, can transmit a lot of bacterial infections, not so much the other pathogens. And birds, of course, are very important because of their viral carrying potential. So if you look at the mammals, for the rodents, the primates, and the bats, you would see that these are actually animals which are in very close contact with humans. Rodents, primates, and bats are almost always found in the peri-domestic environment, in very close contact with humans throughout Asia, in fact, throughout the whole world. You know, if you go out, you will see rodents, a lot of primates, or long-tailed macaques, for that matter, in Malaysia or in Asia. The macaque groups are very prominent. And of course, bats, the fruit bats and the insect-eating bats are very prevalent in the peri-domestic environment. And this is very interesting because these groups of animals, which are high on the zoonotic potential list, are actually the animals which are in closest contact with humans. I've just very quickly listed out the viruses which are known to be transmitted by wild animals, and also the mode of transmission in Asia. This is by no means comprehensive. It's just a snapshot of some of the viral pathogens that we can find in this region. Of course, especially in Southeast Asia, JE, chikungunya, dengue, Zika are very prominent. Then, of course, we've got rabies, avian influenza, and herpes virus, which is very cosmopolitan, not only in Asia, but also in other parts of the world as well. And as you can see, quite a bit of these viruses are transmitted by mosquitoes or by arthropods. Some of the emerging ones are more through direct contact. And of course, you've got rabies, which is through bites of infected animals as well. Looking at the bacterial pathogens, there are quite a number of these pathogens which can be transmitted by wildlife, especially the rodents and the small mammals. Of course, scrub typhus, rickettsial pox are very common in Southeast Asia, and they are arthropod-borne. These are arthropods, mites or fleas or ticks which are infecting rodent and small animal hosts. A very important bacterial zoonosis is from reptiles and amphibians. It's Salmonella, transmitted by the fecal oral route. Leptospira, as you know, is a waterborne spirochete, which is shed by rodents in their urine, and this is very common in Asia. Tuberculosis is very common, as well as Campylobacter. So this is just a snapshot of the bacterial pathogens which we can find. As I mentioned, the list is not exhaustive. There are many more bacterial pathogens which I'm sure you know of, and you can give examples of. The parasitic list is long, and in Asia, as you know, we are a hotspot for parasitic infections. There are loads of parasites, protozoa, helminths, arthropods in Southeast Asia. a large number of them have got zoonotic potential. We look at malaria. Malaria is very rampant. Cystosis, blastocystis, cryptosporidiosis. These are all intestinal and waterborne pathogens. This is a tissue parasite, sarcocystis. Toxoplasma is a tissue parasite. And then, of course, you've got your other worms as well, which are foodborne, and they are carried by various types of wild animal hosts. Today, I would like to focus a little bit on malaria, especially zoonotic malaria, as that is my main focus of research now. And after giving you a snapshot introduction about the zoonotic potential of wild animals, I would like to focus a little bit more on zoonotic malaria. So malaria, as you know, is caused by the parasite from the genus Plasmodium. It infects a very wide diversity of vertebrate hosts, mammals, reptiles, amphibians, birds. They all can get infected by Plasmodium. It is transmitted mainly by hematophagous arthropods, like mosquitoes, for example. In our case, mainly Anopheles and Culex. But of course, you can get infections by leeches as well, especially for the fishes, the amphibians, and the reptiles. They can get malaria infection from infected leeches. But for the terrestrial vertebrates, it's mostly by mosquitoes. Now there are a number of species which can infect humans, and these are the classical human malaria parasites, which is Falciparum, Vivax, Malariae, and Ovale. But now we have got the fifth human malaria parasite, which is Plasmodium knowlesi, and it is recently been recognized as a very important zoonosis in Southeast Asia, and the infection is spreading upwards toward mainland Asia, and also southwards in all the small islands in Indonesia as well. Just a very brief historical timeline on this zoonotic malaria in Southeast Asia. The infection is not new. In 1899, it was already discovered that Asian primates can harbor this pathogen. And then subsequently, in the 1930s, Knowles and Dasgupta described the zoonotic Plasmodium knowlesi for the first time among long-tailed macaques in Malaysia. These are primates. Long-tailed macaques are primates which are very common in Southeast Asia. So they described for the first time the primate from these macaques, and they were actually able to harvest the blood from these infective macaques and experimentally inoculate humans. You know, in those days, it was very ethical to inoculate humans with various types of pathogens to look at the zoonotic potential. A lot of prisoners were volunteered or forcefully volunteered into these experiments. And of course, nowadays, you can't do that. And then subsequently, in 1964, the first case of a natural zoonotic knowlesi infection was reported. And more recently, Baldev Singh from Sarawak actually showed that there was a very large foci of zoonotic malaria among humans in Sarawak. Sarawak is on the island of Borneo. It is a Malaysian Borneo. And about almost 60% of the human infections there were not due to the classical human malaria parasites, but it was due to the new emerging Plasmodium knowlesi. So what is the scenario today since 2004? Now we know that Plasmodium knowlesi is very widespread in Southeast Asia, and it is the most common form of human malaria in Malaysia, and it is also emerging to be the most common form of human malaria in other parts of Southeast Asia as well. This is a list of the Asian primate Plasmodium and the risk of zoonosis. We know that Plasmodium knowlesi is a confirmed zoonosis. Interestingly now, there's a new Plasmodium called Plasmodium cynomolgi, which has recently been confirmed to be a natural zoonosis in Malaysia. That was confirmed a few years ago, and now we are seeing a lot of human Plasmodium cynomolgi infections in Malaysia and also in other parts of Southeast Asia. So nowadays, we are not only dealing with Knowlesi, we've got another guy on board, which is cynomolgi. And the third potentially zoonotic species is Plasmodium innui. And the zoonotic potential is very high for this species as well. However, so far, there are no natural human infections, although experimental infections in the past has proven that this species is of a high zoonotic potential. So these are all the Plasmodium species of primates in Asia and in Africa, and these species are all found in Asia. Right, so what are we looking at the present moment? We see that Plasmodium knowlesi infection is very rampant throughout the whole of Southeast Asia. The infection is high in humans as well as in the macaque hosts. We see the infection was first detected in Sarawak. This is Sarawak here in the Malaysian Borneo. This is peninsular Malaysia. We found infections all over the place now, in Singapore, in Sumatra as well, and infections have been found in Cambodia, Thailand, Vietnam, Laos, Myanmar, and recently it is traveling upwards towards Bangladesh, India as well, and I wouldn't be surprised if it will encroach into China fairly recent soon, because the primate reservoir host has got a very wide distribution, and we also know now that it's not only the long-tailed macaque which can be the host. There are other species of primates as well which can contract the infection from the long-tailed macaques, and these primates are more widespread in the continental Asia. Now, as I mentioned, we are not only talking about Plasmodium knowlesi, we are seeing emerging infections with Plasmodium cynomolgi in Malaysia, in Cambodia, in Sabah, and also in southern Malaysia as well, and in Thailand. So Plasmodium cynomolgi is also taking stage now as an emerging zoonotic malaria in Southeast Asia. Now, Southeast Asia, as I mentioned, is a hotspot for zoonotic malaria, and it apparently is now our biggest export commodity. It is no more oil palm, it is no more rubber, it is no more tin. It is zoonotic malaria, which is our biggest export commodity, because we have exported recently this infection to many parts of Europe. We have opened branches in America as well, and recently there's an export of this pathogen to Australia. So this is not only a localized problem or a localized disease, it is a traveler's disease now. So a lot of travelers coming into Southeast Asia for recreation or for travel or for business, if they go to the forest and if they get bitten by these Anopheles mosquitoes, which are harboring the sporozoites of Plasmodium knowlesi, they may get infected, and it is becoming an emerging traveler's disease in Southeast Asia. And now in 2018, recently, we have exported cynomolgi as well. So this is another commodity which is on our global export list. So let's talk a little bit about Plasmodium knowlesi. As I mentioned, it is the most common primate malaria in Southeast Asia and a very important zoonosis. It's got a very wide spatial distribution in many Southeast Asian countries. The main reservoirs are the macaques and also the leaf monkeys. The main vectors are the Anopheles mosquitoes. And I just like to stop at this point to elaborate a little bit about this forest Anopheles mosquitoes. Traditionally, a lot of the human infections can be traced back to some activity in or around forested areas where the Anopheles mosquito thrives. Yeah? And of course, the macaques, the monkeys, are in the forest, and the mosquito vectors are there. And therefore, a lot of human infections were traced back to forests. And there were very little peri-domestic infections or urban infections. But later on, I will show you a new data which kind of contradicts this, because we are seeing a lot more urban infections now, and I'll try to explain why we think we are seeing more urban zoonotic malaria infections. So as I mentioned, a lot of cases can be traced to activity in or near forested areas. And the macaques or the long-tailed monkeys appear to be asymptomatic. A lot of animals that we have sampled do not show any clinical signs of infection. When we do a blood parasite investigation, we find that the parastemia is very, very low, sometimes even undetectable by microscopy, and therefore they serve as very efficient reservoirs. So they do not get infected, but they harbor low titers of the parasites, and therefore they can persist as very efficient reservoirs. Because through evolution, you would realize that the natural host and the natural parasite would have co-evolved to an equilibrium whereby the pathogen would not kill the natural host. Because if the pathogen kills its natural host, then it itself would die off. So a lot of wild animals are natural hosts to a lot of pathogens, salvific pathogens, and they live in harmony or in equilibrium with these pathogens as well. And as I mentioned, Knowleside malaria is now the 5th human malaria parasite. This is a graphical snapshot of the infections in Malaysia, in humans, malaria infections in humans in Malaysia. And if you draw attention to these orange bars, you would see that these are indigenous human malaria infections, okay, caused by the classical human malaria parasites. The dark blue bars represent imported malaria species, and of course in green you can see the emerging zoonotic malaria caused by Plasmodium knowlesi. So you would see that from 2018, since active surveillance was carried out, the prevalence of zoonotic malaria has increased dramatically to a point that it is now about 70% of the human malaria infections that we see in Malaysia. So a large proportion of human malarias are now due to zoonotic malaria, which is carried by the long-tailed macaques and vectored by the Anopheles mosquitoes. Now, the big question is, if this infection is so common, is it a new parasite, or has it been around for quite some time? We believe that it is not new. We believe that it's been tragically misdiagnosed for other human malaria species, because in the past, the gold standard for malaria detection was through microscopy. But now, with the advent of molecular diagnostic tools, including PCR, we are able to differentiate the species of malaria parasite. But traditionally, microscopy was the gold standard, and it was very difficult to differentiate Plasmodium knowlesi from the other human Plasmodium species, because under the microscope, the trophozoites of knowlesi look very similar to the trophozoites of Falciparum. The band forms of knowlesi look very similar to Malariae band forms and very difficult to distinguish. The schizons look very similar to Malariae as well. You've got all the stipplings in there, and the gametocytes look very much like Plasmodium malariae gametocytes. So in the past, it was very convenient to call the infections Plasmodium malariae infections because everybody thought that knowlesi was not common. But now, as we know that knowlesi is becoming very prevalent in the country, most microscopists would report it as P. knowlesi, P. malariae infections, right? And then it is only confirmed after molecular diagnostics and sequencing of the amplicons. So what does knowlesi do in humans? A lot of infections are fatal in humans with a lot of organ involvement, especially the liver. There's metabolic acidosis as well. Parastemia is very high. In fact, it is a parasite which can produce one of the highest parastemia in all the malaria infections. and the body temperature can go up very high as well. And this is a very interesting feature because in the past, before the advent of penicillin and the like antibiotics, Plasmodium knowlesi was actually used to treat what's that word again? Is a spirochete, neurosyphilis. So what they would do is that people who are infected with the neurosyphilis caused by the spirochete treponema, they would actually inject the patient with Plasmodium knowlesi, and Plasmodium knowlesi would elicit a very high fever, and that high fever would actually kill off the treponema, and then they would get rid of the neurosyphilis, and then they would use some more traditional drugs to kill off the Plasmodium knowlesi. So very interestingly, before the advent of antibiotics or penicillin in particular, Plasmodium knowlesi was the choice of treatment for neurosyphilis. And it also has got a very short erythrocytic life cycle among all the primate malarias. And as I mentioned, the infection is very silent in the primate host with no apparent clinical signs, low peristhemia, and they are very efficient reservoirs. So these are the primate hosts which we know are natural reservoirs of Plasmodium knowlesi and also Plasmodium cynomolgi in Asia, mostly the macaques. But if you look down the list, you will find that apart from the common suspects, you would see that other species of monkeys as well, which are also prevalent throughout mainland Asia, in China and in India, they can get cross-infected with Plasmodium knowlesi as well. So they may not be natural hosts, but they can certainly get cross-infected. And here lies the big problem about the global spread of the disease, because we know that a lot of these primates have got overlapping home ranges, and they are very sympatric in their habitats, and therefore they can very easily transmit the infection from one species of monkey to the next, and therefore the infection can spread very quickly in the whole of Southeast Asia where the monkey hosts are present. Right, today I will focus a little bit on the long-tail macaques because they are the most common natural hosts of Plasmodium knowlesi and Plasmodium cynomolgi. Now these macaques are very abundant and widespread in Southeast Asia. They are very common in the cities and the peri-domestic environment. They've got a very broad home range, a very broad plasticity in their requirements as well. They feed on almost anything. They are in very close contact with humans in the peri-domestic environment, in wildlife reserves and in parks as well. And they have been introduced to many Asian countries out of their natural range. This is the natural range of Macaca fascicularis, or the long-tailed macaque, which is the host of Plasmodium knowlesi. As you can see, it is distributed throughout Southeast Asia. And this is the model for the range of Plasmodium knowlesi. As you can see, it is within the range of the long-tailed macaque, but it also goes beyond the range of the long-tailed macaque up into mainland or continental Asia as well. So now this is the big question mark over here. We know that there are no long-tailed macaques in this region, but there are other species of macaques there, and therefore the theory is that there's a very strong crossover between the natural hosts and these new hosts, which are now potential reservoirs and vectors of zoonotic malaria, which they can then transmit the infection up north into the Asian mainland as well. So this is something which is going to be very interesting to look at, how the infection spreads from the natural host in Southeast Asia up to new hosts through crossover contamination, and how the infection eventually goes up into mainland Asia. So researchers in this area need to be very active in surveillance to really start picking out zoonotic malaria infections. So this is a snapshot of where we are now in terms of the prevalence of infection in the monkeys. We find there's a very high foci in Sarawak, in Malaysian Borneo. Almost 90% of the primates there are infected with Plasmodium knowlesi. In West Malaysia, where I'm talking from now, 1/3 of the macaques are infected, and this is the prevalence list as we go to other Asian countries. The numbers, as you can see, are from some time ago, a few years ago. There's new data now showing that a lot more infections are prevalent in the other countries. And I will give you an updated prevalence rate as well through our study on the epidemiology of Knowlesi in Peninsular Malaysia. So we screened over 1,005 macaques from various habitats in the country, from the various age groups as well. And we did a molecular detection to find out the prevalence of Plasmodium knowlesi and the other zoonotic primate malarias as well. And what we found is that Plasmodium knowlesi is present in about 13 to 14% of our monkeys, our long-tail macaques. Plasmodium cynomolgi is there at about 30%, and this is very interesting now because cynomolgi infections, as you know, is an emerging zoonosis in Southeast Asia, and this is probably the parasite which we need to be very concerned about in the future because it's highly prevalent in the primates, and it has been shown to be able to naturally infect humans as well. So looking at the habitat variables, this is something very interesting. As I mentioned to you previously, most of the human infections can be traced to activity in the forest, and it was therefore believed that the primates in the forest were the most infected with the parasite. But what we have shown is that the highest infections are no longer in the forest anymore. The highest infections are seen in the primates in the urban environment, in the agricultural areas, and also in the suburban environment. So the infection is no longer contained in the forest primates. Primates in the urban environment are highly infected already, and it is only a matter of time when the forest Anopheles mosquitoes will learn to adapt to the agricultural and the suburban areas, and eventually in the urban areas. And once we start seeing these Anopheles mosquitoes, this specific Anopheles species of mosquitoes in the peridomestic environment, then we believe that this zoonotic malaria will start to be a full-blown urban zoonosis. Now, looking at the age groups, we find, of course, as expected, the adults are, the infection is more prevalent in the adults, and the males, being males, always want to dominate the females, so the infections are higher in the males as well. Now, this is a snapshot of the infection among humans in Malaysia, okay? Now, this is prevalence associated with activity in the various habitats, okay? Human activity in the various habitats. You see in the green bar, human activity in forested habitats, and the orange bar, human activity in plantations, and of course, human in the urban environment. Now, what we are seeing just over a year, or across 2 years of data from the Ministry of Health Malaysia, we see that human infections associated with forested habitats is on the decline. But what we see is human infections associated with plantation activities are on the incline, as well as those associated with humans in the urban environment. So this ties in very well with our primate data, which shows that the primates in the urban environment and in the plantations are now more infected with the zoonotic malaria compared to those in the forest. So I'm still crunching the numbers from 2017 up to 2020 now, and trying to see if there's a constant trend to show that there's a correlation between the increase in the primate infections in the paradomestic environment and also the increase in the paradomestic infections among humans. This is some genotyping data which I'd like to share with you, and bear with me, I'm almost done with my presentation. This is a genotyping based on the CSP gene of Knowlesi. Now what I want to share with you is the haplotype distribution data. So we managed to fish out about 25 haplotypes in our limited survey. But very interestingly, you can see that the bars in blue represent the haplotypes in the monkeys, and the red bars represent haplotypes found in humans. Now, two things come up from this. Firstly, we can see that there's a very prominent haplotype in both the monkeys and also humans, and we are doing some more work to determine if this is a virulent haplotype. The second thing that comes out from this is that all human haplotypes are shared with the monkey haplotypes. There are no unique human haplotypes as such. Of course, although the data is still preliminary and we need to screen a lot more monkeys and a lot more humans, but what we have shown now is that it is still a zoonosis. There is no apparent human-to-human transmission, because if there was human-to-human transmission, you would expect some form of genetic drift, and then you will start seeing some unique human Plasmodium knowlesi haplotypes as well. But of course, this data is preliminary, and we need to do a lot more before we can draw any conclusive inference from this. So in conclusion, there's a very high prevalence of P. knowlesi and P. cynomolgi among long-tail macaques in Malaysia and in whole of Southeast Asia. The infection is no longer restricted to the forests, but we are seeing a lot more urban and suburban infections in both monkeys and in humans. We really need to expand our epidemiological investigations, not only in Southeast Asia, but as I mentioned, in the northern part of the Asian mainland. Researchers need to be vigilant. They need to be surveys carried out to see if the infection is spreading northward. We need to look at other primate species to see if they are effective reservoirs of infection, and how the long-tailed macaques are cross-infecting other species of macaques as well, which can be found in India and in China and in Nepal and in Bangladesh. There's a very serious threat for tourists in Southeast Asia because we are a hotspot of infection. Now, this is also a big challenge for the control of malaria, because the macaques will be a constant source of human infection as long as the mosquitoes are present, and therefore malaria elimination in Southeast Asia and eventually in Asia is going to be a very big challenge, because the salvetic cycle is always going to be there. There is almost no way for us to get rid of the macaques in the forests and also in the paradomestic environment, and therefore malaria elimination is going to be very challenging in the future. This is a snapshot of the risk of malaria in Asia. We are looking at areas with low risk or with high risk of infection. But this is very interesting. This is a projection for malaria elimination in Asia, in Southeast Asia in particular. And we can see that, you know, Malaysia is supposed to be malaria-free in 3 years or so. Other parts of Indochina are supposed to be malaria-free in less than 3 years. And many countries in Southeast Asia are embarking for malaria-free status as well. But this is based on the classical human malaria parasites. But now, with the advent of zoonotic malaria, this is not going to be a reality. There is no way we are going to see malaria-free countries in less than 3 years, because zoonotic malaria is now 70% of the human malaria infections that we are encountering. And very timely, this paper was published in the Malaria Journal 3 years ago, which says that the World Health Organization needs to really focus on zoonotic malaria, because this is currently the biggest threat in Southeast Asia. And we need to initiate a surveillance program among range countries and in other neighboring countries as well, to look at the distribution of the infection. We need to understand the salvetic cycle in more detail, and we certainly need to approach this disease in a One Health way, looking at epidemiologists, veterinarians, medical doctors, primatologists, sociologists, all coming together to solve this common problem. And of course, we need to take into account the agent, the host, the environment, and the vectorial factors which are in this epidemiological quartet as well. So with that, I want to say that the increase in zoonotic outbreaks is really a symptom of a broken relationship between humans and its natural environment. And this situation is going to worsen if we do not do something about habitat destruction, if we do not do something about global warming, and we must be very proactive. I think the SARS, the COVID-19 disease has shown us that wildlife can be potential reservoirs of global pandemics, and habitat destruction is a very important key driver to address when it comes to human pandemics as well. So I'd like to thank my group at the university, in my lab, all the students who did all the fantastic work. I just sit there and get to present the data, but they did all the hard work, trapping the primates and doing the surveys. I would like to thank my colleagues in University Malaysia Sarawak, especially Professor Balbir Singh. I would like to thank my colleagues in the Wildlife Department Malaysia for their support in carrying out the studies. Of course, University Putra Malaysia, where I'm from, and also my colleagues in the University of Malaya, who have contributed a lot in this study, especially in the human aspects of zoonotic malaria infection. And before I end, this is my last slide, I promise. I just want to share with you a very exciting conference which is going to come up in 2021 in October in Kuching, Sarawak, which is the global hotspot for zoonotic malaria. Okay, no, it's not anymore, but I'm sure in 2021 it'll never be. So in Kuching, that's where we're going to have this conference. It's a very beautiful state. There's a lot of culture and heritage, a lot of fantastic food, and it's very cheap. And the conference is going to be organized by the Federation of Asian Veterinary Associations. It is an event which happens every 2 years, where all the Asian veterinary associations get together and have the Congress. And in 2021, the Malaysian Veterinary Association will be organizing the Congress in Kuching, Sarawak. So please go to our website, fava2020.com. All the information is there regarding the program, the speakers, the registration fees, and also a lot about Kuching, Sarawak, and why you should join us at the conference. So thank you all very much for your kind attention, and I will be very happy to answer any questions if there are any. Thank you very much.
Thank you very much, Professor Reuben, for enlightening us with burning problem of zoonotic diseases, especially during this COVID-19 time. And we are learning many things about the zoonotic diseases. So you may have some questions. Before handing over to Dr. Anis, I am getting some questions. What steps to be taken to prevent rabies in wild animal?
Okay, thank you very much for that interesting question. And the reason I say it's interesting is because it's also a very challenging question. It's something which has been on the minds of rabies researchers for a very long time. As you know, rabies has been around for a very long time. It is almost impossible to control the salvific cycle of rabies because there are many wild carnivores which are efficient reservoirs. And this includes the civets, the wild cats, and of course the wild dogs. And even the bats are very efficient reservoirs of the infection in the salvific cycle. Then, of course, you get the spillover to the domestic animals, the domestic dogs, especially, and also cats. And then it's very difficult in the first place, it's difficult to do surveillance program in forest animals because it takes a lot of effort and expenditure. And what we found recently is that you can get serological titers of the rabies virus, but it's often very difficult to isolate the pathogen. So we do not know for sure whether these reservoirs are actively harboring the virus or whether it's the titers are due to previous exposure. So yeah, it's a very challenging thing, and I'm not sure If anybody else in this forum can enlighten us on how we can lessen the rabies burden in wild animals, but as far as I know, no one to date has come up with anything conclusive as of yet. Yeah. So I'm not sure if there are any more questions. If there are none, then can I say thank you? Or
Dr. Anis, is there any question?
Yeah, there is one question. What is the status of malaria vaccine across the globe?
sorry again, Professor, I can't
Status,
educate you.
yeah, status of malaria vaccine.
Oh, yeah. So the malaria vaccine, as you well know, is in trials now. There are some clinical trials on the malaria vaccine. But of course, again, it's very challenging because, you know, the malaria parasite expresses a number of epitopes on its surface. And the vaccine can potentially only cover certain epitopes. So yes, clinical trials up to now has shown some promise. But I'm not sure if it's going to be a pan vaccine for all the human malarias, and certainly not for the zoonotic malaria because none has been developed for Norasa yet. But certainly it appears to have limited efficacy in controlling malaria, especially in areas where the infection is very high. I know that we do not routinely do this in Southeast Asia, but I guess in Africa and in South America, there is some trials on the malaria vaccine which is showing some promise. I'm not sure in India if there is trials with malaria vaccine. Maybe somebody can enlighten us.
Hello? Hello?
Hello, yes.
Yes, sir, I'm audible? Am I audible to you? Hello? Hello?
Hello, yes, we can hear you.
Okay, am I audible to you, ma'am?
Yes, yes, we can hear you.
One more question, sir. What do you think malaria-free world? What are the approach?
What's an approach for a malaria-free world?
Yeah.
Oh, well, to be honest, in the past, there was a light at the end of the tunnel for a malaria-free world. but now, with the advent of zoonotic malaria, the light is becoming quite dim. Or even if there's a light at the end of the tunnel, it's probably the light from an oncoming train. And so we're not sure how this zoonotic malaria is going to pan out. We are not going to be able to suppress the salviatic cycle for sure, because the population of primates are too high. And as long as the primates and the mosquitoes are there, we will be seeing zoonotic malaria. But certainly for the classical human malarias, many countries are heading towards malaria-free status, and that is certainly possible, but with the classical human malarias, not with zoonotic malaria. The only way to get rid of zoonotic malaria is to kill the human host. Then there won't be any more zoonosis. But unfortunately, I don't think killing the human host is an option. So, so I think we will be stuck with zoonotic malaria for quite some time.
Thank you very much, sir.
Thank you.
Now, may I request Dr. Anish to give his the concluding remarks of the webinar along with vote of thanks. Over to Dr. Anish.
Thank you, ma'am. Good afternoon, everyone. I, Dr. Anish Yadav, a professor at SCOS Jammu, is hereby presenting vote of thanks. It is a matter of great privilege for me to express the vote of thanks on my behalf as well as on the behalf of the organizing committee for the successful conduct of the international webinar on zoonotic diseases and novel approaches in worm control. First and foremost, our heartfelt and sincere thanks to Professor J.P. Sharmaji, Honorable Vice Chancellor, SCOS Jammu, and Chief Patron of the webinar for his inspiring inaugural address, though he could not deliver it today because of some busy schedule. Sir, despite COVID pandemic, you are always at the forefront in promoting the university activities and scientific discussions at various platforms. Sir, once again, we thank you for the mentorship. I may like to express our sincere thanks to Dr. J.R. Rao for kindly accepting our invitation and putting the opinion remarks on the field of parasitology. Sir, your benign presence and guidance is always a source of inspiration for all the parasitologists, especially young generation across the country and the globe. Thank you, sir, for being with us. We have had an extremely useful day. We are extremely indebted to both the guest speakers. We are grateful to Dr. Chandra, ma'am, the Chief Editor, Tropical Biomedicine and former Director of Research and Innovation, Malaysia, for sharing her experiences of a scintillating career devoted for upliftment of the farmers, especially dealing with small ruminants. Your today's talk and work on sustainable management of worms affecting livestock is a benchmark for the researchers not only in Asian continent but also across the world. Once again, I thank you, ma'am, for gracing the occasion. Our sincere gratitude to Dr. Reuben, Professor Veterinary, Laboratory Diagnostic, University Putra, Malaysia, for enlightening us about the role of wildlife in zoonosis. Sir, your talk will be a guiding force for all the researchers, academicians, and policymakers for considering wildlife as one of the important component in combating the zoonotic diseases. Sincere thanks, Dr. Reuben. The man behind this show is definitely our esteemed Dr. Rajesh Katos sir, Dean Student Welfare, SCOS Jammu, and Head of the Division Veterinary Parasitology, who as the patron was the motivating force for the conceptualizing and conduct of the program. Sir, we thank you for all your support and encouragement. Our sincere thanks are also for Dr. M.S. Bhadwal, Dean Veterinary Faculty and co-patron for this webinar and his unstained support in organizing this webinar. My sincere gratitude to Dr. Shanku Borkataki, organizing secretary of the webinar, who left no stone unturned in making the day to happen. Thank you, ma'am. The driving force behind the organization of the show is definitely the participants of the webinar. The participants were very enthusiastic. I am thankful to all the participants for attending the webinar. My thanks also go to all the people who have given their precious time in organizing the webinar. In particular, I would like to acknowledge and thank Dr. Rajesh Godara, co-organizing secretary, for compilation of the technical compendium, Dr. Amandeep Singh for managing the website, and Mr. Sindhu for making IT-related arrangements. We also thank press and electronic media for the excellent coverage and publicity being accorded to the event. Also, the people who may have not been mentioned here but have directly or indirectly helped in organizing the fest and this webinar are also being acknowledged. Thanks again to one and all again for gracing the occasion. Thank you very much.
Thank you very much to all of you.
Yeah,
Organized.
ma'am, it's our pleasure.
Thank you.
Thank you very much. Very well done webinar.
Shanku, ma'am, please. Sanku ma'am, I think her connection is not there. Please wait. Sanku ma'am,
Thank you. Hello.
yeah, ma'am, continue.
Okay. Thank you, Dr. Anish. Now, I thanks to all the speakers, participants on behalf of organizing secretary, and I declared the webinar is over. Thank you, ma'am.
Thank you. Have a
Thank you,
nice
ma'am.
Thank you. Have a nice day.
Okay, congrats.
Okay, sir. Thank you, sir.
Thank you, Dr. Sanku.